Okay. Good afternoon, everyone. Wildlife is something which man cannot construct. Once it is gone forever. I am Supriya Naika hosting this program. I welcome one and all for the 82nd episode of Creative Online Talks. Wildlife is a major part of the ecosystem. The environment balance can never be maintained without wildlife. This conservation of wildlife is very much necessary for us. To save the earth, we need to conserve wildlife. Different factors bring a threat to wildlife. To spot light on this, Creative organize a uh, talk on unsung heroes. What do herps to do for us? To present this, we have Dr. Dayatima Ghosh with us today. Before start, I like to brief about Creative. Creative is a group of people who are like-minded, who are eager to gain new knowledge. Creative vision is to create to build constructive thinking on various domains, and it main focus on non-textual, non-academic, non-syllabus related concept. Creative is a platform to the subject experts to present their views, subject related discussions on the Creative platform. I thank all the resource person for their time and their knowledge for sharing on Creative platform. Every weekend we organize online talks on Zoom platform, and we live streaming on our YouTube channel Creative GBD. I call Raghavi to introduce today's speaker. Over to Raghavi.
Good afternoon, everyone. I am Raghavi, Creative Volunteer. It is my pleasure to introduce today's speaker, Dr. Dayatima Ghosh, who is a postdoctoral fellow at Ashoka Trust for a research in ecology and the environment and visiting researcher in University Lincoln, UK. Ma'am completed her bachelor's degree in zoology honors from Maulana Azad College, University of Kolkata, and master's in zoology from Kolkata with specialization in genetic and wildlife conservation. Ma'am received her PhD from the University of Kolkata on ecology in 2021. Ma'am worked on how agriculture impacts on reptiles and amphibians and their ecosystem functioning. Today, ma'am will be sharing their insights on why we need reptiles and amphibians, at what pace we are losing them, and why should they have to be conserved. I hope you find the presentation both informative and inspiring. Thank you, Creative, for giving this opportunity. Thank you.
Thank you, Raghavi, for the beautiful introduction. Good afternoon, all. I'm Dayatima Ghosh, and today I'll be talking about reptiles and amphibians. They're often referred to as together as herpetofaunal. Now, these are very mysterious creatures. They are there in the background, but we hardly see them. We don't know what they're doing. They're not as noisy as the birds or conspicuous as the mammals. Hence, they pass unnoticed, and we do not know about their role in the ecosystem. It's still a mystery. So today we'll be talking about what these animals are doing in our farmland, what are we doing to them, and why, if at all, we should be conserving. But before that, we'll take a look at ourselves. At present, we are 8 billion, and we are only increasing in number. With this increase in number, we are also increasing the demand of agricultural produce. And to meet this demand, to satisfy this rise in demand, we are converting natural habitats into agricultural land. With the increase in demand, there's also increase in the negative effects of agriculture, one of which is evidently seen in the loss of biodiversity in and around farmland. But this loss is not just losing a few species or individuals, it's losing something beyond that. It's losing the ecosystem service. What are these services? These are the benefits that we receive from the farmland, from the natural resources and the nature free of cost. But if we have to imagine how much is this benefit, You can put a monetary value which accounts to hundreds of trillions of dollars. There are four major forms of services. One is the provisioning. Nature provides us with food and water. There is supporting function where all the species interact to maintain the ecosystem functioning. There are regulating services like pollination, pest regulation, and simply aesthetic value. Nature always gives us pleasure. So we wanted to know how this increase in agriculture is impacting these reptiles and amphibians in farmlands. We conducted a study in Orissa Balasore district. And the photograph in the right panel, you can see the different stages of degradation, habitat degradation. The first one is a farmland with natural vegetation around. The second one, or the middle one, is a degraded forest. And the third one is an extremely intensive farming with no natural vegetation nearby. So what we did was we divided the sites into low and high intensive farming. So continuing, like in the next slides, we will be using these color codes. Low intensive farming is highlighted in green circle, and the high ones are outlined in yellow. So low intensive farming means there's less pesticide input, there is less fertilizer application, less agricultural land use, and very high proportion of natural vegetation. On the contrary, high intensive farming means high pesticide application, high fertilizer, high area, proportion of area under agriculture, and lesser to no natural vegetation. So the next challenge was, how do we survey these animals? Because they're cryptic, we do not know what they do, where are they? So we use a combination of methods. One is passive trapping, I'll be discussing shortly. The second one is active trapping. So the small yellow rectangles which you see on the right photograph on the right side are the cover boards. These are simple structures, rectangular structures of either wood or even leaves. We just leave them in the field and they act as artificial refuge because these animals always seek for shelters, always seek for refuges. So we just add on the number of refuges for them to come and take a shelter. In the next morning, you go and flip them over, see whatever individuals are there, take the data and release them back. It's called active trapping because you're not capturing any animal, but you're simply adding a refuge in their habitat. The third one was transect walking. We walked for 250 meters and we collected all the reptiles and amphibians that were present that we could spot. And finally, active searching. So the red circle in the left photograph, it covers an area of 5 hectares. We actively searched this area for any and every reptile that we could spot. Now, what are these passive trapping sessions? They're also called as drift fences, and they look like this, the photograph on the left side of the screen. They're basically polythene sheets which are erected in farmlands, and the end of it is buried inside the soil so that no animal can escape through it. Now, this fence basically utilizes the behavior of these reptiles and amphibians. Whenever the amphibians and reptiles get obstructed by a barrier, in this case, the drift fence, they do not attempt to cross it. Rather, they start following the trail. And along this trail, we had our traps, pitfall traps, which is the photograph at the right bottom. These are 20 liters buckets, which are dug within the soil, or double-ended funnels, which are simply cylinders with 2 funnels fixed at the 2 ends so that if any animal enters, it cannot come out. Now, as they get obstructed by these fences, they start moving along the trail and they end up in one trap or the other. The next day you go, take them out, take the measurement, and after that release them back in the wild. So how bad was the impact? These were the list of reptiles and amphibians we observed in the field. You can see that the abundance of reptiles and amphibians was extremely low in the high intensive farming, which you can see in the yellow bar, compared to the green bar, which represents the abundance of these herpetofauna in low intensive farming. But even though these reptiles and amphibians are together, often together referred as herpetofauna, but they are very different in their behavior and their physiology. So we wanted to see if the response was the same for reptiles and amphibians. In amphibians, you can see that the abundance is extremely low in high intensive farming, again, by the height of the yellow bar, comparing the height with the green bar, which is the abundance in low intensive farming, where there was lesser pesticide, lesser fertilizer, and more natural vegetation. But when you see the same graph for the reptiles, the abundance is no doubt less in the high intensive farming, but it's not significantly less compared to the low intensive farming. They are, of course, responding, but the response is not as prominent as that of the amphibians. Does it mean amphibians are more vulnerable? We'll come to that point shortly. Next, we wanted to see what about the species in the low and high intensive farming? Are they different? They are not. So if low intensive farming had A, B, C, D, E species, the high intensive farming also had A, B, C, D species. Essentially, the composition was the same. So the first question is, why were the reptiles not responding as much as the amphibians? Maybe because they were less vulnerable, or they were going through something called an extinction day, which means it's a lag phase between the onset of the effect of intensification on reptiles and the time the reptiles need to react to that. To give an example, if today I start eating a lot, I will not gain weight overnight. I'll gain weight over a certain period of time. This period of time is the lag phase. Reptiles need that lag phase, the time phase, to respond. Maybe we will see the response in the coming years. There were three very interesting outcomes from this study, which are very sad. One is the species composition is the same, as you can see in the plot. The species richness was the same, and most of the species we found were generalists. They were generalists in using the habitat. They were not very specific. Now, these three findings indicate the fact that whatever we have, both in low as well as in high intensive farming, they are the climax species. If we continue doing what we are doing to our habitat without taking active step in conservation, we might be losing all of them from our farmland. Now, we need to conserve these animals, but how do we do that? Agricultural land does not mean only agriculture. It has got a lot of other components. For example, water bodies, human settlements, plantations, fallow lands, natural vegetations, semi-natural vegetations. Each of these components are called land use. So we wanted to know how these reptiles and amphibians respond to various land use and agricultural system in an increasing distance from the farm. For that, we color-coded this different land uses. The green one is the natural vegetation. And if you look at it compared to the agriculture, which is in yellow, you can understand what we have done to our habitat. This photograph itself says how much we have destroyed our habitat and the level of land transformation. These agricultural lands literally have minimum to no natural vegetation around them. The only patch of green that you see is the forest area. Anyway, continuing further, we classified the landscape into plantation, water bodies, human settlements, fallow lands. What are fallow lands? Whenever you go to a farmland, you can see there are many lands which are just left barren. There is no agriculture, nothing happens there much. It's because the soil could be very rocky, or the soil is very hard. So these are just left like that, but these are very important, and these are the favorites of the reptiles. So after classifying all the land use components, this is how the study sites look like. The concentric rings that you see around the study sites are the increasing distance from the farmland, from the 5 hectares farmland where we sampled to 1 kilometer or 1,000 meters. The ring, which is the outer ring, is at the maximum distance of 1,000 meters, then 500, then 250, and then 125 meters from the farmland. Simply by looking at these two plots, you can tell that even within the same group, that is amphibians, abundance and richness depend on different components at different distance from the farmland. I left you all with a question of why amphibians were more vulnerable. Maybe we have an answer here. So you can see that the reptiles depend on AII, which has pesticide as one of the major component. So it depends on pesticide at 125 meter distance from the farmland, which is understandable because the farmers are applying pesticide. So the effect can be seen in the nearest landscape scale. But what about these amphibians responding to pesticide at 1,000 meters landscape scale? The second point to note here is the water body. Amphibians are biphasic. They depend on water as well as on land to survive. And water is a fantastic source for contamination of pesticide. It causes soil leaching and takes the pesticide farther away from the farmland. This is one of the reasons why amphibians are more vulnerable than reptiles. The second could be amphibians are very less mobile compared to the reptiles. Maybe in a lifetime, an amphibian moves 1 to 2 kilometers or even less, depending on the size of the amphibians. The third reason could be because of their permeable skin. Now, amphibians can also breathe through the skin. and they have to go into the water, which is extremely contaminated, which means there is direct entrance of pesticide inside the body. Reptiles are pretty much safe because they are scaled animals. The only way they come in contact with pesticide is through inhalation or through food which contains particles of this pesticide. This could be the reasons why in our study, amphibians were more vulnerable than reptiles. But coming back to these two plots, if I am a conservation biologist and if I want to improve amphibian abundance, if I go and suggest that increase natural vegetation, which is abbreviated as NV in these plots, beyond 1,000-meter landscape scale, that conservation action has to fail because amphibians might not even travel that much distance in their lifetime. Similarly, for reptiles, you can see the richness and abundance depend on different land use at different spatial scale. If I want to improve the diversity, and if I say increase semi-natural vegetation, which is SMV here, immediately surrounding the farmland, that conservation action will not be successful because they depend on natural vegetation at a further distance from the farmland, at 500 and 1,000 meters. These informations are fundamental when we are trying to conserve an animal. We need to know which animal is benefited from which particular land use in which landscape at what distance from the farmland. Only then can we successfully end up conserving animals, not only reptiles and amphibians, but this is applicable for any and every animal in our farmland or in our habitat. So now we know that agriculture is reducing the herpetofaunal diversity, especially amphibian abundance. But what use are these amphibians? They are insectivores, which means they feed on insects. And a lot of this insect in the farmlands are crop pests. So basically they function as regulating services. They provide us regulating services of reducing the crop pests. But as we are losing the abundance, as we saw in the plots, how does it impact their functioning? So for this particular study, we looked into three important components. One is frogs, a super predator, then arthropod predators, which are beneficial. For example, dragonflies, which feed on the crop pests like grasshoppers. So we wanted to see the interaction between these three in a paddy field. So to do that, we conducted a large-scale experiment covering 7,500 square meters. We established this black-colored boxes or plots of 100 square meter. They had paddy, and they were covered with mesh so that we didn't have any birds coming and feeding on the frogs or the crop pests. In the left side, you can see there are three colored boxes. Again, coming back to the same color code, green stands for low intensive farming, where we had 10 frogs per 100 square meter. Yellow stands for high intensive farming, where we had 5 frogs per 100 square meter. And the gray boxes had 0 frogs, which are the control plots. So we wanted to see how this pest and beneficial arthropods grow in presence and absence of different densities of frogs. These were the field events. We started with land preparation, followed by experimental unit preparation, followed by seedbed preparation for paddy. Then we collected frogs and maintained them in a natural habitat and fed them every day. Then we transplanted the paddy seedlings and released the frogs. But how did we quantify the pest and the beneficial arthropods? That is the grasshopper, for example, the grasshoppers and the dragonflies. We did sweep netting. These are basically nets which are used to capture insects. And later on, we preserved them and we identified them. We also did night sampling, where we went on looking for the frogs, if the frogs in the experimental plots were doing good and were alive. And we did stomach flushing of the frogs to see what all food do they eat. Results were something fascinating. So we will focus especially in S3, which is the sampling phase 3, where paddy has reached its maximum vegetative growth. It has got the maximum number of leaves and tillers. So you can see at that stage, in absence of frog in the control plots, the ratio of pest and predator were 1:1, which means for every pest predator, for every dragonfly, there was 1 grasshopper available. Something was different when we added 10 frogs. The pest abundance somehow was increasing. Something bizarre happened when we had 5 frogs. Pest population shot up even more and the predator abundance decreased. But this wasn't supposed to happen because We thought amphibians would be feeding on the crop pests. At the same time, we had these beneficial arthropods like dragonflies who would be feeding on the crop pest. Hence, there would be an increase in overall pest population in my farmland. But something was not right because there was something going on called an intra-guild predation. Here, frogs and beneficial arthropods had a common prey, which were the crop pests. What we did not know was frogs also feed on these beneficial arthropods. So when the density of the frog is below a threshold, for example, 5 frogs per 100 square meter, the intra-guild predation was very high. We have to consider 2 things here. One is the behavior of these animals. Now, frog, this particular species, is an ambush predator, which means it just sits and waits, scanning movement for prey. Whenever it scans that there is a movement in the prey, it hops on and feeds on it. So movement is an important cue. Now at S3 phase of paddy, when paddy has a lot of foliage and leaves available, crop pests being very cryptic in nature, they start taking refuge within the leaves, within the crop plants, with least movement. So even when the crop pests are present in your site, literally they are invisible because their movement is less. Comparison to that, beneficial arthropods, the dragonflies, they're extremely agile, they keep on moving and flying, and they become easy to detect. So if I am a frog and we are only 5 in number, and we see an easy target in front, we do not need to go and seek for the crop pests. We will simply feed on the beneficial arthropods because I feed both on arthropods plus crop pests. For me, it doesn't matter. So what am I doing is, 5 of us are only targeting the beneficial arthropods. As a result, we are avoiding the crop pests, and the crop pests are increasing in abundance. But again, since we are feeding on the beneficial arthropods, the predation pressure of these arthropods on the crop pests are reduced. As a result, crop pests shoot up even more. And rather than providing an ecosystem service, when the density of frogs are less, they start providing us with an ecosystem service, which we cannot afford at this point of time when human population is only going to increase and there is no stop to agriculture. We lose almost 40 to 60% of crops to crop pests. We cannot afford that these beneficial animals start providing us with a disservice. This necessitates why we should start conserving these animals. But how do we conserve them? Habitat management. We need to know on which land uses these animals depend, which land uses are beneficial, and where to exactly conserve them. At what distance from the farmland should we conserve them? Now, for the next few minutes, I'll be talking about animals from a different perspective, from an aspect where animals, especially reptiles, are capable of learning, capable of understanding the changes in their habitat, in their surrounding, processing the changes, deciding, learning, remembering, and finally acting on them. From an aspect where these animals are cognitively skilled. So we know a lot about mammal behavior, our behavior, but do we know enough about the reptiles? Are they simply stupid, dumb creatures who are instinctive and always ready to bite you? They are not. So I worked with Calotes versicolor or oriental garden lizards. You can see them in your backyard as well on the fences basking. They're extremely abundant in croplands. They're less vulnerable than amphibians, as we just learned. They're highly adaptable. They can adapt to certain changes in their habitat. And the females are excellent at reproducing. They produce 17 to 21 eggs in one clutch. They're diurnal, that is, they are active during the day. At night, they sleep like this on the leaf blades. Again, they're sit and wait foragers. That is, they sit in one place and scan for the movement in the prey. Once they get it, they jump and they feed on the pest. And they are cognitively skilled. They can learn. But can they? Let's see. We wanted to see if these reptiles can learn about crop patch, which has more pest abundance. And if they're at all capable of learning, how does that impact their pest consumption? So this study I did in University of Agricultural Sciences, Bangalore. This is how the field site looks like. It's basically a patch of forest land. And we converted a small patch into a cropland, which you can see on the right side of the screen. It's been brinjal crop. So in this system, I had oriental garden lizards as the predators. brinjal as the crop, and crickets as the defoliators. Now, they are defoliators because they feed on the leaves of the crop plants, and as a result, the crop plants die. There's no crop. You can see the small hole this insect has made after 2 minutes of re-releasing them in the experimental plot. So they are pretty much a very severe pest. So what did we do? There are 8 lizards performed in this experiment. Each of the lizards performed for 10 trials and each trial lasted for 30 minutes. Let's see. So each of these lizards have this plot which has orange flags. The orange meant that that particular patch had more pest density. You can also see the crickets hopping around. The colorless patch had very less pest density. So the position of this orange patch was kept constant for each lizard. We gave them enough time to learn about the position of the infested patch. And over the 10 trials, we explored if they could make correct choices. By a correct choice, we mean that they chose to enter the orange patch at first compared to the non-colored patch. The second, we wanted to see how they allot the time between the orange patch and the colorless patch. And if they at all learn about this position and the reward that they're getting from the orange patch, how does that impact their pest consumption? For a farmer, this would mean how that impacts the pest regulation. We'll just see this lizard catching a prey. It's already eating one. So this is an ambush predator. It just sits and waits for a prey to come. And you can follow the eye movements. The eye movements are actually for the movement, scanning for the movement of the prey. We'll just give it some time. Yeah, you can see, yes, right. So what did we find? Now remember the color orange. For the first time, we got to know that lizards do make correct choices, and the number of correct choices they make significantly improve their pest consumption. You can say that the lizards learned about the position of the orange patch by the significant number of correct choices it made. What about time? The more time the lizards spent in the orange patch also improved their pest consumption, and they learned to spend more time in the orange patch compared to the colorless patch, where the pest was less. But how did this learning impact their pest consumption? Pest consumption from the orange patch was double that of the incorrect patch. Over very less number of trials, these reptiles were capable of understanding the difference between the two patches based on prey available, and they decided to act on it. They are that intelligent. Now imagine what they would be doing in the field. In a cropland with differently infested crop patches, reptiles would learn which patch is more rewarding with more pest available, would frequent those patches much more, would spend more time, and would help in suppressing crop pest population at least double than that of the less infested crop patches. Next, we wanted to know if the lizards do discriminate between taste of prey and if they can associate color learning with taste. So we gave them food in this green bowl. Then we gave them food in a green and a blue bowl. Now blue was the unfamiliar color, it was the new color. Then what we did was something wicked. We just changed the quality of the prey in the green bowl. So the green, which was rewarding till now, was giving them a very bad food. So we wanted to know if they were capable of understanding that green is no longer good and reform an association with blue color as a rewarding food. What did we find? This is how we did the experiment. These are the enclosures where we kept the lizards. This is the green bowl with a bad food. It's a blue bowl with a good food. And this is the third trial of this individual. Here comes Gabby, she's a female. And she goes to the blue bowl at the first. She completely ignores the green bowl. She has learned over 3 trials that green, which was a familiar color, which gave me good food, was now giving me very bad food. And I do not want to go to the green, but blue is my preferred color. How did it affect the other behavior? Latency here means time taken, right? So when there were 2 colored bowls with similar food, you can still see that the reptiles took some time, more time to go to the unfamiliar color that is blue over the green. This difference in time was not very significant, but still it shows that they were trying to decide whether to go or not. See what happens when we switch the taste of the prey. The green and the blue line are flipped over. Now they were going to the blue bowl much faster compared to the green bowl, which indicates that there is a chance that they were hesitating to go to the green bowl. They were trying to decide if the green bowl will give me good food, because last time it was pretty bad. This is an example where we can see that they were unlearning that green is that green is rewarding. They were relearning that now blue is good, green is bad. And how did that impact their prey consumption? Prey consumption was pretty much the same when both green and blue had the same quality of food. Because I'm a lizard and I simply look for movement in the prey, any food given, if good in quality, I don't mind differentiating between the color. But when green is bad, you can see that they started rejecting the green. They started taking food only from the blue. And this happened over only 3 trials. Lizards were capable of unlearning an association and relearning, reforming a new association with taste and color. Therefore, they have their own mind, their own intelligence, their own sophisticated cognition, which allows them to perceive the changes that happens, to discriminate between the cues which are available, to decide and finally to act on them. Till now, we have known that applying pesticide, which is one of the major consequences of increase in agriculture, it results in destroying our biodiversity, which is much more. We're also losing the ecosystem services. What if we start considering the predator intelligence, the predator cognition, not only of reptiles, of birds, amphibians? We can do this experiment with any beneficial animal that we want. And by harnessing on this, by integrating this cognition and their intelligence, we can improve their prey specificity, thus improving overall biological pest regulation. Once this is done, we will be more reliant on nature and natural resources and less dependent on the chemicals. If this is achieved over a certain time period, we will be improving the biodiversity in and around farmland. And once we do this, all this biodiversity will start adding on to the ecosystem services. We convert our system into a sustainable system where we are not only more reliant on the nature and the natural resources, but we also conserve them. This is what our goal should be. Because we are at a point, if we continue doing this, it's not long that we are going to lose all the beneficial animals. We know that agricultural intensification reduces reptiles and amphibian diversity, especially for amphibians. They're more vulnerable. However, these animals are excellent in providing ecosystem service like biological pest regulation. There could be a lot of other more, which I did not address here. But however, we have to maintain them at a particular threshold below which we are not only going to lose the service, but these animals will be turning against us and performing this ecosystem disservices, which we are in no place to afford right now. And these animals have excellent cognitive power. They have their own mind, intelligence, they can learn, remember, and so on. They have their own rights to survive, not only because they're beneficial to us, but because they're amazing animals and they have full rights to survive along with us. With this, I'll end up in my talk, but I'll show a few photographs from my field. This is the drift fence in a highly intensive farmland. The left top photograph is how we used to install the drift fences. The left bottom are the double-ended funnels. We had to construct all these traps because they're not available in India, and we could not import them from abroad as well. So this took almost 6 months for us to make the whole setup, and there were 3 of them, 3 of these whole setups. The right one is a pitfall trap. These are 20-liter buckets, which we had to bury inside the soil. This is how we did the active searching. These are the beautiful 9 reptiles who performed all the cognitive tasks and the behavioral tasks. Thank you so much. I'll be very happy to answer your questions. Thank you.
Thank you, ma'am. There is one question in chat box.
Okay, sure. I'll stop sharing my slide.
Based on your experience, do you suspect any difference in behavior of herpetofauna? 1 minute. Forest and agriculture lands.
Oh, excellent question, Vikram. I am sure there should be. I haven't looked into their behavior in the forest and in the agricultural land. But yes, that would be an interesting study to do. But from my experience of working with these lizards, I think there should be some. Thank you for the question. Yeah.
Srinath sir.
Thank you, madam. It was a good talk. Coming to the what you call interaction with the farmers to establish your experimental study, how difficult it was, because generally people don't allow it. If it is in GKVK, specifically labs you will make outside, but in the outside, that is first question. How difficult is to engage with the farmers to make your experimental facility? Second thing is in both the herps like lizard or other things, frog. What is the, what type of blind beliefs or myths are associated when you have gone through various cultures either in Karnataka or West Bengal? Just can you share some?
Yeah. Hi, sir. Thank you. Brilliant questions and very important. So I did a work with farmers in Orissa as well, but because of the shortage of time, I didn't share. But now I'll speak a little about it. So when I initially started working on reptiles and amphibians, there was a kind of a disbelief because they know that they're just there in the farmland, but as I mentioned, they don't know what they do. And as we started working within their farmlands, thankfully, all the farmers actually allowed us to do what we did because we were not interested in their crops or destroying the land. We were just seeing what animals are there. So initially, it was fun for them that these lady and these people just come and I don't know what they do in the farmland and they just go back in the evening. But as time passed and in the 3 years of this work, we could see their attitude change. They would come early in the morning and wait near my trap, wait for us to come to see what all are there in their field. Now, this is a big achievement. Those people who were just not bothered about what's there in the field, except that the crop pests are destroying their crops and they need more crops, were now actually curious to see what animals are there. Also, there were 2 different groups of farmers. One, as I said, were not at all interested in what was there. But there was this other group who knew that these animals helped them in bioregulation. I did not need to tell them at all. They knew it. But their point of view is, what do I do? Because I have to grow crops, I have to feed my family. I understand what you're saying is very important, but I need an effect which is very quick. I cannot wait for the reptiles and amphibians to function for 1 year. I need a benefit which is quick, and pesticides do give them that benefit. So when you start working with the farmers, the first thing is you have to build a connection. It's not that they do not know what's there. They know a lot more than me and you because they grow with the system. They literally are a part of the system. We are outside. But you just need to tell them that, see, this also happens. See, this animal is intelligent enough to do it. And they're pretty fascinated because they don't have the time to think, oh, the lizard might be remembering the crop pest. They don't have the time, honestly. But as outsiders, as conservationists, as ecologists, as scientists, researchers, or even just volunteers who are working for conservation, we just need to go and tell them. We just need to make them a little more curious. This was the first question. I hope I answered it. The
Yes,
second, so the second question was about some meats. There are a lot. So again, there are groups of farmers who kill any and every snake that they see, whether it's venomous or non-venomous, doesn't matter. It's not a myth, but it's a conflict. And that's one of the reasons why we are losing the reptiles. For them, it's just that, yeah, one snake has bitten my brother who died, so I killed all the snakes. But maybe that snake was a cobra or something else. But the one which you killed yesterday was just a checkered killback. It helps you in your farmland. For them, for us, it's very difficult to address this because here we are actually conflicted. We are talking about conservation versus we're talking about people's life. This takes a lot of time. And also coming to the myth, the Oriental garden lizard, which I worked with for the behavioral study, they are in Orissa, they are referred to as Roktachosha, which I'll just try to translate, which means bloodsuckers. Because they look down, their behavior is they look down from the tree like that, head down, and the males have orange head. That's because that helps them to make themselves conspicuous to the female while they're breeding. Breeding males have that orange, beautiful head. And they think it's orange because it looks down and it comes and sucks blood from your forehead, so you die. There is no point conserving these animals. To these farmers, when I go and speak and when I tell them, see, this is not happening, this is what they do, this is what they're doing to you, I'm sure that's going to benefit. I'm really hopeful that the farmers do appreciate.
Yeah, thank you, madam. Thank you.
Thank you.
One more question, ma'am. Chat box.
Yeah. Based on your field experience, what different do you see in your select different species of reptiles and amphibians in your study, like different species of toads or different species of reptiles? Hi, Dipanjan. Thank you for the question. I'm not sure if I understood the question properly. Can you please tell me once? Is it related to the behavioral study?
You can unmute.
Hi, can you hear me?
Yeah, I can. Yeah.
Hello.
Yeah, I can.
Yeah, hi. Good evening. First of all, thank you so much for this vivid presentation. Now, I just wanted to ask that during your field study, you said that you've selected 5 toads, okay, 5 frogs. So just I wanted to know, I'm curious about knowing that, is that if you select 5 different species of frogs, okay, so is there any difference that you find in consumption or in behavior of this species during your study? Or maybe like in 1 year you have selected maybe one species of frog, okay, but in the coming year or like coming 6 months you find 5 different species of frog. So do you find that there is any difference in your study result from this one species to the second species? I'm just curious about it.
Yeah, brilliant question. Thank you for this. So the 5 frogs that we worked on with for the experiment was based on the density. Right, so we found 5 frogs in 100 square meter in the high intensification plot and 10 frogs per 100 square meter in the low intensive farmland. Now, this particular frog had a particular feeding rate. They will be feeding on 3 crop pests in probably half an hour. Like the lizards, they feed on 8 to 10 crop pests in 30 minutes. Your question of having 5 different species, yes, absolutely, it's going to change the experiment because different species would have different rate of feeding. It also depends on individual status, but if we are talking about species, definitely it will vary. We could not bring the species into the experiment because it was becoming extremely elaborate and complicated, and complication because our knowledge is extremely limited. So we try to use a native species of frog, which is abundantly found in agricultural lands. Because if I'm also talking about bioregulators, I need to have a high threshold, as I mentioned, because below that, they will be performing as disservices. Therefore, we got stuck with one species. Yes, in the next year, we find a lot of different species. It may not happen that fast, but if it does, then yes, the pest regulation will definitely change. So if one species can do this much, now imagine if 10 species are there in your farmland, what can they do? And therefore, you should conserve your species again. I hope I could answer your question, Dipanjan.
Yes. Any more questions? Ma'am, what you are doing in your post doctoral projects, ma'am? What?
Yeah, so the postdoctoral project was about this reptile cognition, where I look into if reptiles can learn, they can discriminate between cues, and how can we actually use them for our benefit to improve their biological pest regulation. So the last 2 studies with the reptiles was a part of my postdoc work.
Any more questions? I'll just chat box. No more questions. Okay. Ma'am, it's a complete any government initiation projects you are working on this type of work, ma'am? Your PhD work on that?
No. My focus has been on the ecological aspects and community outreach. I haven't really interacted with the government.
Any work with the government like this is going on? Any ideas, ma'am?
Honestly, no. Because so we have this biological pest regulation, which focuses on the beneficial arthropods, but it never comes to reptiles and amphibians. No, not any that I am aware of.
Okay. Okay. Any more questions? Vikram sir. Okay. One minute, ma'am. You muted.
Hello.
Vikram
Good
sir.
evening, I'm Ramesh from KSTA.
Sir.
It was a very good presentation by Madam. Congratulations. I have one question, Madam.
Yeah,
Hello.
sure. Yeah, sure. Hi.
Did you find any difference in productivity of paddy when you stock less number of frogs and high number of frogs?
Yeah, thank you. Again, an excellent question. We did not find any difference in the paddy yield. This is obvious because 5 and 10 are extremely low in number. This also raises a question that we say low intensive farming is better than high intensive farming. But my study shows that it isn't, because right at this point, whatever is present in the low intensive farming is extremely low. Therefore, in that graph, if you noticed, there was an increase in pest population as well. 10 and 5 frogs definitely are not doing any good to our habitat. Whatever we have in low intensive farming is much lesser than the threshold that we need to improve the bioregulation, hence the crop yield.
Could you find out the threshold number of frogs that could be stocked in an acre of paddy field?
Yeah, so I did not do it, but there are reports which say at least 150 frogs, that to bullfrogs per 100 square meter, can effectively bring down crop pests and improve the paddy. Now, reaching 150 per 100 square meter is pretty unrealistic right now. I am not sure of the exact density, but this requires a lot of more experiment with varying density.
Correct. Okay, madam, it was a very good presentation. All the best for you, for your postdoc.
Thank you so much.
Thank you. Supriya, madam, thanks.
Thank you, Ramesh sir. Thank you.
Supriya, madam, can I ask one question?
Yes, sir.
Yeah,
Vikram.
Dr. Ghosh, very nice presentation.
Thank you.
Just wanted to ask, what kind of diversity of species overall did you find in the agricultural fields of both amphibians and reptiles, of all herpetofauna together?
Yeah, I'll just.
Are there any patterns in that? Thank you.
Not any distinct pattern, but yes, I can share this slide, which has the list of reptiles and amphibians. Can you see that?
Yes, we can. Yeah.
Yeah. So there were 14 species of amphibians and around 8 species of reptiles. But these species were pretty much common in both low and high intensive farming.
Okay.
So there were common Indian toads, which are pretty much everywhere. There were 2 toad species. There were balloon frogs, then bullfrogs, which are supposed to be very good regulators of pests, crop pests. And in the skittering frogs, which are mostly found in the water bodies in the agricultural lands, they're also supposed to be very good bioregulators. Then in the reptiles, there were oriental garden lizards, which were most abundant, and hence we did the behavioral study with them. There were also rat snakes, which are pretty good at regulating rats, which are severe crop pests of the stored grains. Yeah, these were the species that we found.
Madam, one information, the previous speaker who has questioned Dr. Ramesh, who is a KSTA chairman, which is inside GKVK campus. Just for your information, I also request Dr. Ramesh to use Madam in one of the similar workshops or conferences. Dr. Ramesh, sir, if you are there.
Yeah, please. Madam's presentation was really very good and inspiring. We regularly organize webinars, Madam. I will be inviting you for a talk one of the evenings.
I would love that.
Thank you, Ramesh ji, because you will be doing a lot of biology conferences, workshops as a part of KSTA. Thank you.
Yeah. Thanks.
I would love that, sir. Yeah.
Yeah. Thank you very much.
Yes. Thank you so much.
I think no more questions. Okay.
Yeah.
Special thanks to Vikram sir, who suggested us for to take your talk, ma'am, and Raghunandan sir. He is always supporting us. Okay, I think we can wind up here. 1 minute. Okay. Thank you for your time and knowledge for sharing with us. It's really a scientific talk, ma'am. It's like your presentation more. No, it's a highly scientific type of talk we had today. We are very thankful for your patience and for answering all the questions and all. Thank you very much, ma'am. Thank you
so
all
much,
the
Supriya.
participants. Yes, ma'am.
My special
I know you are very.
Madam, my special compliments to you and Srinath sir. You always have been inviting excellent presenters, and talks are really very useful for students and general public. Thanks.
Thank you, sir. Thank you, sir. It's okay, ma'am. Shall we wind?
Yes. Thank you so much, Supriya, for getting in touch. It has been amazing. Thank you everyone for listening to the talks and all the brilliant questions.
I know, ma'am, you are very busy from.
No, I am not.
I know, but still you are very good. Thank you, ma'am.
I love doing this, Supriya. Thank you so much.